Hello everyone and welcome to Breaking Science. It's time to think like a scientist because, as you probably noticed, there's a global pandemic going on. But thanks to a Herculean effort from a number of scientists all over the world, we've got several potential vaccines. Some of these vaccines are what we call mRNA vaccines, which are different from the childhood vaccines that you're probably familiar with. The purpose of this video is to share with you some background about what mRNA vaccines are, as well as some specific information about the first two vaccines for coronavirus that have been granted emergency use authorization by the FDA of the United States, made by the pharmaceutical companies Moderna and Pfizer. Importantly, what this video is not going to do is tell you whether you as an individual should or shouldn't get a particular vaccine. That's always best handled as a discussion with your doctor who knows your particular medical situation. However, I hope the information contained in this video can form part of an informed decision about the use of RNA vaccines. The first section of this video is about mRNA vaccines in general, how they're different from more traditional vaccines, and what we know about their safety from other mRNA vaccines that have been in the clinic already. Next, I'll cover what we know about the design, safety, and efficacy of the Pfizer and Moderna vaccines. I won't discuss the upcoming AstraZeneca and Johnson & Johnson vaccine or any of the others. Although these are likely to be made available to the general public in the near future, they are not mRNA vaccines and probably deserve their own video. Finally, I'll discuss the unknowns about the Pfizer and Moderna vaccines and when we're likely to have these open questions cleared up. mRNA vaccines are still an emerging field, but the information that I'll share in this video comes from high-quality sources, peer-reviewed scientific literature, clinical trial data, as well as FDA reports. I'll share links to the evidence in the description of this video. I also want to declare that I have no conflict of interest with respect to the subject of this video. With that, let's get on to mRNA vaccines. In order to understand what a messenger RNA or mRNA vaccine is and why someone would make one, we first need to take a journey inside a cell and review some basic cell biology. Deoxyribonucleic acid, or DNA, encodes everything that makes up you. It's the instructions for making all the proteins that build your body, but it's just the blueprints. DNA is hidden away in the nucleus of your cells, and in order to do anything with it, it must first be copied into ribonucleic acid, or RNA. This molecule goes out into the cytoplasm, where it's processed into mature mRNA. Messenger RNA, or mRNA, is basically useless for anything except telling your ribosomes what proteins they should produce. It can't integrate into your genome and doesn't survive long in the cytoplasm. Proteins are made in the cytoplasm and then shuttled wherever they need to go. They can be found inside the cell, attached to its surface, or secreted out. Importantly, RNA is normally only found within the nucleus and cytoplasm. If it leaves your cell, it is very quickly broken down by enzymes called ribonucleases. Traditional vaccines are either weakened or killed versions of the virus or bacterium you're trying to protect against, or their proteins, subunits as we call them, derived from the infectious agent. mRNA vaccines deliver the messenger RNA molecule to your cells and get them to produce the protein themselves. As I mentioned earlier, RNA floating around outside your cells is broken down very quickly. If you were to inject mRNA vaccines directly into a patient, they would likely not do anything, as the RNA would be degraded very rapidly by endonucleases floating around. As a result, RNA vaccines are packaged in lipid nanoparticles. These are essentially very small balls of fat that do two things. They protect the mRNA from enzymes that would chop them up, and they help the message get into your cells where it can be useful. Nanoparticles have been used in drug delivery for years. Notably, they've been used to deliver chemotherapeutics more specifically to tumors in order to limit some of the toxicity associated with systemic delivery of this class of drugs. Every cell in your body is surrounded by a lipid membrane, so lipid nanoparticles don't look foreign to your body and are quite safe and well tolerated. Over the years, we've learned how to modify these nanoparticles to control where they go or increase delivery of their payload into cells. I won't cover all of these modifications, but there's been a tremendous amount of work over the years controlling lipid nanoparticle delivery. I've included a link to a paper in the description that covers the myriad of clinical trials going on using lipid nanoparticle delivery of drugs. One of the interesting things about mRNA vaccines is that they don't stick around in your body. Although the pharmacokinetic data have not been published for the COVID vaccine specifically, we know that lipid nanoparticles are rapidly cleared by the liver. Even if any mRNA were to leak out of these nanoparticles, it would be degraded within minutes. And the message that's delivered into your cells is also transient. Natural turnover of RNA is quite fast, and because the RNA in mRNA vaccines is mature messenger RNA, meaning it has a poly A tail and a cap, there's absolutely no chance that it could integrate into your DNA permanently. It's a safe assumption that the vaccine itself and any proteins made as a result of it would be cleared from your body within 3 to 4 days, although the data have not been made publicly available specifically for the coronavirus vaccines yet. mRNA vaccines were first conceptualized in the 1990s when they were tested in animal models of multiple infectious diseases. These models included diseases such as rabies, Ebola, Zika, and HIV. In many of these models, the vaccines provided good protection against the disease, and the vaccines themselves were well tolerated. This sparked a number of phase 1 or phase 2 clinical trials run by different companies, including Moderna, starting all the way back in 2006. The protection afforded in the case of the HIV vaccines was unimpressive, but there are at least very promising signs of immune activation for rabies and flu. However, more relevant for our story today is that in these trials there were no common unexpected adverse events. This was the first proof of concept that mRNA vaccines could be safe to use in humans. Within the past 15 years or so, there's been intense interest in making vaccines for cancer. Unlike vaccines for infectious disease, which are given before you're infected with the infectious agent, cancer vaccines are generally given therapeutically after the cancer has already begun. Not surprisingly, many of these vaccines were mRNA vaccines. No less than 59 clinical trials have been initiated for mRNA vaccines for the treatment of various types of cancer. The success of most of these vaccines in clinical trials could be described as moderate at best, and as a result of the lack of protective effect of most of these, they have not been granted FDA approval. However, each of these trials has further contributed to validating that the mRNA approach is safe in humans. A common point expressed by skeptics of the mRNA vaccine platform is that mRNA vaccines are too new or untested, and it's certainly fair to say that we have a lot more data about traditional protein or pathogen-based vaccines, which have been dosed in millions upon millions of patients over the years. However, even prior to the initiation of coronavirus mRNA vaccine trials, mRNA vaccines had been dosed in thousands of patients, and the data showed that these vaccines were largely safe. We should ask ourselves how much data we need to convince ourselves that something is safe. Remember, we're really trying to prove a negative here, that vaccines don't cause harm. And there's no experiment that you could actually design that would conclusively prove that. Instead, it's important to consider the risks of taking a vaccine against the risk of getting the disease that you'd be vaccinated against. This brings us to the coronavirus vaccines themselves. At the time of making this video, there have been 2 vaccines that have been granted emergency use authorization by the FDA of the United States. One made by Pfizer and BioNTech, and one made by Moderna. Let's dive into what each of these are made of. The Moderna vaccine, which was originally called mRNA 1273, encodes the spike protein stabilized in a pre-fusion state, meaning that the spike is forced to look how it does before the virus actually enters your cells. This helps ensure that the immune response you're generating is primarily against the entry receptor before it's been used. If you're trying to keep Batman out, you can think of this as blocking the bat grappling hook before it's actually attached to the building. The lipid nanoparticle in this case is based on polyethylene glycol, which you can find in skin creams, toothpaste, personal lubricants, and as an anti-foaming agent in many food and drinks. The exact formulation, of course, has not been revealed because of intellectual property law, but there is more detail in the FDA report. This vaccine was tested preclinically and found to protect animals from coronavirus by causing production of anti-coronavirus antibodies, a common feature of vaccine responses. It's important to note that in these models, the animals were actually challenged with coronavirus after being vaccinated, which is something we can't ethically do in humans because we have no cure for COVID. Animals that were vaccinated were protected from upper and lower respiratory tract damage mediated by the coronavirus. These findings and others form the justification to proceed with clinical trials in humans. Pfizer and BioNTech initially advanced 4 different vaccine candidates into phase 1 clinical trials in both Germany and the United States. Interestingly, BNT162b2, the candidate that ultimately received emergency use authorization in the United States, is also an mRNA encoding pre-fusion stabilized spike protein, encapsulated in polyethylene glycol-based lipid nanoparticles. Conceptually, the Moderna and the Pfizer vaccines are actually pretty similar. The Pfizer vaccines had similar preclinical promise, resulting in antibody production. This graph actually shows some phase 1 clinical trial data where different doses of the vaccine induced different amounts of antibody. So these 2 vaccines are pretty similar in makeup, and both had promising preclinical phase 1 and phase 2 activity. These trials are proof of concept showing in a relatively small number of people that an investigational drug is safe. What did we find when these vaccines were advanced to larger scale phase 3 trials? A note on emergency use authorization. This is a mechanism meant for public health emergencies such as the current pandemic, where the public is given access to a promising drug that hasn't yet been granted traditional FDA approval. This does not mean the drugs are untested. To the contrary, the bar for safety and efficacy is still quite high. Both Pfizer and Moderna had to get initial data from phase 3 trials with many thousands of participants each in order to apply for emergency use authorization. Beyond these data, to get EUA, a company needs a plan for active safety follow-up, including monitoring deaths, hospitalization, or any other adverse events that may occur. So it's important to keep in mind that we will continue to get more data about the safety of these vaccines as time goes on, although the results are unlikely to change dramatically from what we've seen initially. Both Moderna and Pfizer set up their phase 3 trials to report follow-up data of 2 months to 2 years after the second dose of their vaccines were given. This 2-month time point for interim analysis, or early data, was selected because we know from decades-long study of vaccine literature that most adverse events that could occur following vaccination happen within 6 weeks. And this time point also gives you an idea that any protective effects of the vaccine could be continuing long-term. The Moderna phase 3 trial was randomized, observer-blind, placebo-controlled, and intended to evaluate the safety and immunogenicity of the vaccine, as well as how well it works. 30,351 individuals were randomly split into placebo or vaccine groups, stratified into classification of people that are 18 to 65 with low risk for severe COVID, the same age group with high risk, or people that are over the age of 65. 82% of the people in this trial were considered to be at occupational risk of contracting the virus and included healthcare and other essential workers. Participants were given 2 doses of the vaccine 28 days apart. If someone in the trial developed any symptoms or adverse events of any severity, they went to their doctor for a nasal swab to confirm infection status. The primary endpoints for this study were mild symptoms or a positive coronavirus test. The secondary endpoints were severe disease, death, or infection more than 7 weeks after the second dose. This study was set up to test how well the vaccine prevented mild or severe disease and is planned to be carried out for 2 years. We got results of interim analysis of this trial in November of 2020, and we were updated with even more data in early December. This corresponded to a time point of a median of 2 months after the second dose of vaccine was given. There were 196 cases of coronavirus in the study, 94% of which were in the placebo group. This suggests that the vaccine is very effective at preventing COVID. Importantly, the vaccine also had very good efficacy in individuals over the age of 65. The blue curve here shows the number of cases that accumulated over time in the placebo group, and the red curve shows cases in the vaccine group. This is another way to visualize the stark protective effect of the vaccine. In fact, the vaccine was shown to be effective in a number of different high-risk groups, such as those with chronic lung disease or diabetes. There were only 30 cases of severe COVID reported among trial participants, all of which occurred in the placebo group, suggesting the vaccine is very effective at preventing severe disease. So we know the vaccine works very well, in fact far better than some people had imagined. But how safe is it? The most common adverse events attributable to the vaccine were low-grade headache, fatigue, and muscle pain. These are common for most vaccines. Severe adverse events, meaning anything that required hospitalization or death, were no more common in the vaccine group than the placebo group. After the release of the emergency use authorization reports, a few vaccine recipients have had severe allergic reactions, which were highly publicized. It's important to note that these individuals had a history of responding to vaccines with severe allergic reactions, and this was successfully treated by doctors. This is a normal but very rare side effect of traditional vaccines. And remember, at the time of recording this video, we've already lost over 340,000 Americans, 1.8 million people worldwide to this virus. Altogether, these data suggest that mRNA 1273, Moderna's RNA vaccine, is not only highly effective but also very safe. As I mentioned, we will continue to get more data in the upcoming months and years, but the FDA granting emergency use authorization suggests that in evaluating all available data, the known or potential risks of getting the vaccine are far outweighed by the known risks of getting the virus. The phase 3 Pfizer vaccine trial was set up very similarly to the Moderna trial, with the same follow-up time point of 2 months to interim data, 2 years eventual follow-up, and similar primary and secondary endpoints. This trial enrolled 43,448 individuals, stratified in 4 groups: ages 16 to 18, 18 to 65, 65 to 75, and 75 to 89. In contrast to the Moderna trial, which was a US-based trial, the Pfizer trial also included hospitals in Argentina, Brazil, Germany, South Africa, and Turkey. Also of note is that the 2 doses of the vaccine are separated by only 3 weeks, compared to Moderna's 4 weeks. There were 170 coronavirus cases in total in this trial, 95% of which occurred in the placebo group. This vaccine also worked well in the 55-plus crowd. Here you can visualize in real time the number of cases in the blue placebo group and the red vaccine group. Importantly, the Pfizer vaccine also had good efficacy in individuals with at least one comorbidity that put them at risk of severe COVID. If we consider the safety data, we see a very similar story to the Moderna vaccine. Low-grade fevers, fatigue, headache, and muscle pain were somewhat common, consistent with other vaccines. The frequency of severe adverse events was low and similar between the vaccine and placebo group, suggesting that the adverse events observed in this study were not attributable to the vaccine itself. So there you have it. The Pfizer vaccine also has very high efficacy and appears to be very safe, similarly to the Moderna vaccine. The documents and data that the FDA reviewed and eventually approved are publicly available on their website and are linked in the comments, and contain a wealth of more details. If you've got further questions, I highly recommend digging back into these documents, as they're quite informative. There are certainly still some things that we don't know about the COVID vaccines. Perhaps the most obvious question is, how long does immunity last? Because it's very hard to determine when someone was actually exposed to SARS-CoV-2 and whether they may have been re-exposed, we still don't know how long immunity lasts after natural infection. Current estimates are that natural immunity to infection lasts at least 6 months. As with many infections, there's likely to be some variability person to person, even in healthy individuals. It's safe to assume that people who are taking immunosuppressive drugs, either for cancer or autoimmune disease, or those that are otherwise immunocompromised, may not have such a lasting protection as healthy people. However, depending on the degree of immune compromise, it's possible the vaccine will still provide some protection. Again, this is a discussion that's best had with your doctor about your particular situation. There's another factor that will influence how long vaccine-derived immunity lasts, and that's the likelihood of the virus to mutate in such a way as to avoid the immune response. Some viruses, such as the influenza virus that causes the flu, mutate very rapidly. For flu viruses, on average, there's 1 mutation every time the virus replicates, and someone who gets the flu and has severe disease could have virus replicating thousands of times inside them over the course of their illness. Most random mutations will reduce the ability of the virus to replicate, but some will increase its fitness. So the virus that the infected person sneezes out at the end of their illness could be quite different from the virus that went into them initially. SARS-CoV-2 generally accumulates mutations half as quickly as influenza does, and a quarter as fast as HIV, so it's a relatively slow mutating virus. Mutations do still happen in coronaviruses and can be driven by the immune response to the virus. We can track some viral variants derived from superspreader events based on small changes in the genome. There was a conference at pharmaceutical company Biogen in late February 2020 that ended up as the root of about 300,000 infections, which we know because the person who spread the virus there had a median of 6 nucleotide differences, 6 mutations that stood out from what we expected the Wuhan viral genome to look like. This particular strain had been found in Europe previously, but not in the US until then. Although we can track very small changes in the viral genome that correspond to only 1 or a few mutations, unless they're in a very unlucky spot, this generally doesn't translate into escaping the immune response. We've also recently identified a new, apparently much more infectious strain of coronavirus in London. It has accumulated 23 mutations, 6 of which actually change proteins that the virus makes. Some of these mutations are in the spike protein, which could explain why this strain seems to be much more infectious than the parental Wuhan strain. It's still unclear whether individuals who are infected with the original strain of coronavirus will be protected from this new strain, although we should get an answer to that question in the near future. A virus that mutates as slowly as SARS-CoV-2 is less likely to be able to mutate to avoid the immune system compared to a virus like influenza. We've characterized about 4,000 individual mutations in the spike region alone, but these haven't tended to occur in the same virus. The London strain shows it's possible to accumulate multiple mutations that change the biology of the virus. Only time will tell, though, how good our immune response is at keeping up with changes in the coronavirus genome. A tremendous advantage of the mRNA vaccine platform is that from a manufacturing standpoint, it's incredibly easy to modify the vaccine to protect against new strains as needed compared to traditional vaccines. Another big question surrounding the coronavirus vaccine will be whether it can provide sterile protection or not. Having sterile protection or sterilizing immunity means that your immune response is so robust that even if you encounter the virus again, it gets blown away before it can replicate in you. A vaccine can still be protective without giving you sterile protection, and indeed most vaccines don't give you sterilizing immunity. In this case, if you're infected with the virus after being vaccinated, the immune response prevents the virus from replicating enough to make you sick, but there's still the potential for it to replicate a little bit in you without any other obvious symptoms. This would be similar to asymptomatic infection. This might sound like an academic distinction, but it has real implications for how we should behave after being vaccinated. If the coronavirus vaccine does not give sterile protection, which is entirely possible and perhaps likely, can vaccinated people subsequently come into contact with an infected person and transmit the virus to someone who's uninfected or vulnerable? This is an open question, but until we know the answer to this, the safest practice would be for people who are vaccinated to continue to wear masks, which we know are very effective at reducing the spread of virus-bearing droplets. Hopefully this video has helped you understand what RNA vaccines are, what we know about the safety and efficacy of the available coronavirus vaccines, and what we still hope to learn about them. Making informed decisions is always important, and these vaccines face an uphill battle against campaigns designed to spread misinformation and confusion. If you decide to share what you've learned in this video with others, I would strongly encourage you to do it respectfully. Asking questions is always a good thing, and although it may feel great to clap back at someone who's spreading misinformation, establishing a relationship of trust and honesty is the best way to get someone to listen to you. Along these lines, if you have further questions or would like to engage in discussion, please do so in the comments section of this video. I'll address as much of this as time allows, but disrespectful comments of any viewpoint will be deleted. If you've got ideas for other topics you'd like me to cover on Breaking Science, please also leave them there. And until next time, stay curious.